Well, good evening. We're absolutely delighted to be in University College Cork for our Academy Discourse this evening as part of the 66th annual meeting of the Paleontology Association. And we would like to thank Professor Maria McNamara for her invitation and for all Maria's incredibly hard work to get this discourse set up and get us all organised. I'm Mary Canning. I'm president of the Royal Irish Academy, and also with me this evening is Academy Secretary Professor Mary O'Dowd, who's in the front row, and joining us online is our Treasurer Professor Patrick Honohan and our Executive Director Dr Tony Gaynor. So if you will just allow me to explain to you, our audience tonight. And it's lovely to see so many of you here on a beautiful summer's evening. And I know that we have about 130 people also who have signed up to join us virtually. Academy Discourses are the oldest and most renowned series of talks in Ireland. The first discourses were presented in 1786, and historically they were the occasion reserved for the most distinguished academics to present their research and their work and reveal their findings to the public. We now record these discourses, and they are all available on the Academy's website. And before we begin, I have a bit of business to transact, which is Academy business. I have to refer to the minutes of our last discourse, which was a fascinating discussion on tuberculosis as a persistent global scourge of humanity, and very relevant again to our times with the recent pandemic. And this was given by Dr Lalita Ramakrishnan on the 26th of May 2022, and it is also posted online. As I have not been informed by any of our members that there were any issues with the minutes of that meeting, I will take them as approved and I will sign them when I get back to Academy House. Our speaker for this evening's discourse is Dr Larisa DeSantis, and she is part of this wonderful conference that you're all about to engage in. And our respondent is Paul Giller, who is a member of the Royal Irish Academy. I'd like to talk about Larisa for a moment. Larisa DeSantis is a Chancellor, Faculty Fellow and Associate Professor in the Department of Biological Sciences and the Department of Earth and Environmental Sciences at Vanderbilt University in Nashville, Tennessee. She studies fossilized mammals to determine their response to ancient climate change. And we talked tonight about the appropriateness of thinking about climate change on today, a day that we have exceeded temperatures in this century. So I think that what Dr. DeSantis is going to talk about will have a resonance for us this evening. She will talk about the potential reasons for mammals becoming extinct and the long-term consequences of climate change and large animal extinctions on a diversity of plants and animals. Dr. DeSantis earned her degree from the University of California in Berkeley, from Yale University, and she earned her PhD from the University of Florida. She is a recipient of the National Science Foundation Career Award. She studies mammals on all continents except Antarctica, and much of her work is explicitly aimed at helping conservationists to understand better ecosystems, past and present. Our respondent this evening is Paul Giller, member of the Royal Irish Academy, who is Professor Emeritus of Ecology at the School of Biological Earth and Environmental Sciences here in University College Cork. So now, Dr. DeSantis, I hand over to you. Thank you very much.
Great. Thank you so much for that kind introduction and for having me here tonight. So I'm going to talk a bit about how we can use the fossil record to better understand what's happening today with climate change. So in the words of Barack Obama, the climate change is no longer some far-off problem. It is happening here. It is happening now. It is happening today. And across most of Europe today, we're having extreme temperatures. So Ireland may surpass its 33.3 degrees Celsius record for the first time since 1887. Further, there are parts of Europe and especially mainland Europe that are dealing with catastrophic wildfires. So what I'm going to talk about today will hopefully resonate with many of you with what's happening at this moment. So what I aim to understand is biotic responses to climate change. How have plants and animals responded to these changes over time? And while most people focusing on this topic are ecologists or conservation biologists, paleontologists have a unique ability to actually provide perspective on modern-day conservation. So I don't want to underscore the importance of ecology and conservation. Those are absolutely critical disciplines. But I will say there is some challenges to it. Because if I want to study, say, this jaguar here, I need to go out, I need to write a grant, I need to monitor populations of jaguars, I need to do this over several years. And if I'm lucky, maybe I get a 3-year grant or a 5-year grant, maybe a 10-year grant to do this. Or if I'm really lucky, then maybe I work with folks to develop a long-term ecological research station, and we monitor plants and animals for the next 100 years, or a series of students do anyways. Well, this is really important information, and we need to continue to do this work. We need to know what's happening today, but we can also take a look into the past and use the fossil record to extend our time axes, to ask both unique questions, but also similar questions to those that we ask today with ecological and conservation studies. Now, a lot of my work focuses on the importance of diet and understanding the dietary behavior of animals, in particular mammals, and how they respond via diet to changes in climate. And the reason why we focus on diet is because diet is absolutely critical to everything an animal does. So for example, koalas are obligate consuming things like eucalyptus. They need forests. Without forests, there are no koalas. Other things like wildebeest are going to move across their landscape differently based on the resources that are available. And it can even influence sort of the movement of biomechanics, right? So how a cheetah captures its prey and takes down this rabbit is going to, what it's eating is going to affect its biomechanics. It can also affect reproduction. So how often an animal reproduces, if it's seasonal, if it's multiple times through a year, and different mechanisms. So diet's really critical. So our lab is focused on dietary reconstructions and ecological assessments of mammals, also known as the DREAM Lab. And most of our work at the beginning was focused on studying these modern animals to understand how we could use them to study the past. But in reality, now what we're focused most of our work on is actually using our understanding of modern animals to study the past, but then studying the past and extracting important conservation lessons that we can actually apply to today. So these are some of the ancient ecosystems that I work, including Brea Tar Pits, which is up here and right here, Australia, and these are two of the places that we're going to spend quite a bit of time in tonight. Now, short of getting in my time machine or using my little time turner here to go back in time, we need proxy methods that can actually infer dietary behavior in the past. And so to do this, we use a variety of different approaches. So morphology, or the shape of jaws and teeth and bones, is really helpful, but that only gives us a first approximation as to what an animal's doing. It tells us, for example, that we are all omnivorous, right? But But in this room, there might be vegans, vegetarians, those that really like fish or sushi, those that prefer red meat. And so that gives us the potential, but it doesn't tell us actually what the animal was doing at that moment in time. For that, we need other methods. And one of these methods is dental mesowear, which gets at the sort of macroscopic shape. Are teeth sharp and pointy? Are they sort of rounded over? Are they flat and worn down from grazing on abrasive grasses? And then we have sort of other proxies that can get at diet as well, like dental microwear texture analysis and stable isotope analysis. And I'll talk more about these in just a moment. So you are what you eat. Everything you eat is incorporated into your tissues, into your hair, into your blood, into your breath, into your fat. Every piece of you is incorporating what you eat. But the great thing about it is teeth and bones are particularly resilient to diagenetic alteration or changes over time. And so we can literally examine the chemical signature locked in teeth, and that can tell us something about what an animal was eating, if it was eating grass or if it was eating browse, for example. So in particular, because plants photosynthesize differently, we can look at these differences, whether we have C4 grasses or C3 trees and shrubs. In places like East Africa, at lower latitude localities, we can use isotopes alone to get at grass versus browse consumption. Now, in other places of the world, it gets a little more tricky. So in Australia, we have C3 grasses and C4 shrubs. We have C4 grasses and C4 shrubs, C3 grasses and C3 shrubs. And it gets much more complicated to actually parse these apart. So we need another method, which is why we use dental microwear. But we can also get at sort of the density of a canopy. So within a C3 ecosystem, like we're in here today, we can get at more open environments or more closed environments. And that can be also really useful to telling us about the ecology of animals of the past. So microscopic wear techniques have been used since the 1970s, and essentially what was done is they would look at a microscopic wear surface using a scanning electron microscope, and then a human observer would go in and count the numbers of pits and scratches that they might see on the surface. Well, it turns out we're pretty good at counting scratches, not so great at counting pits. Is this one pit? Is this many pits? How do we perceive those pits? Now, fortunately, the method has evolved, and we now no longer count these features. We rather analyze a surface much like analyzing sort of a topographic map in 3 dimensions. Now, the reason I have a picture of the Last Supper up here is because that's what it's getting at, essentially the Last Supper, the last few weeks, months, or maybe year of the animal's life, depending on the animal. So 2 of the main aspects of microwear that we look at are, one thing is called anisotropy, which is the degree to which features of similar relief share similar orientation. So essentially getting at sort of the scratchiness of the surface. And what we find when we study modern animals, which we observe them eating, is we see that things like bovids or kangaroos have very high anisotropy. We also see things that are folivorous, eating tough foods like folivorous sloths or giant pandas also have high anisotropy, and things like cheetahs, which eat lots of flesh, also do as well. Now, complexity is getting at surface roughness at different scales. So essentially, if you are in outer space and you look down at the Earth, it looks very smooth. And the closer and closer you get to the surface of the Earth, the more complex that surface is going to get. You're going to start seeing mountains and valleys and topography. Well, we know that relationship is always going to exist, but we can actually measure the slope of that relationship, and that slope is our complexity value. And from looking at modern animals, what we can find is things that are eating insects, things that are eating hard fruits or hard seeds, or processing fruits that have hard seeds, or eating lots of bone have high complexity values. So now that we have these tools, we can then position ourselves to ask broad questions, including how has climate change affected mammalian communities and their floral environments in the past? Specifically, what are some of the consequences of past climate change? What are the consequences of the extinctions that have happened on survivors? And also, what cautionary lessons can we apply to conservation today? So one of the places that I spend a lot of time doing my work is in Australia. And fundamentally, we can use Australia a bit as a canary in the coal mine. They are facing some of the most extreme aridification that this world is seeing. You're seeing fires that are happening that have never happened in the same intensities. You're seeing frequent droughts. You're also seeing frequent flooding events. So the climate is changing rather dramatically. We can look at the fossil record in Australia and also begin to understand the impact of aridification in the past. And one of the places we can do that is a fossil locality known as Cuddy Springs. So before I go any further, I just want to mention that it's really the work of Dr. Judith Field, who has worked on this site for decades and carefully excavated it, and all of her work, her foundational work to determine the age of the specimens, that's allowed us to do the work that we've been able to do since then. But essentially, Cuddy Springs is a site that has different ages represented. So there's a fauna that's approximately 400,000 years old, and another part of the site where it's about 30 to 40,000 years old, and evidence of humans and megafaunal remains together. So we can actually look at this to see what sort of climate changes were happening between about 400,000 years and about 30 to 40,000 years. So one of the ways we can do this is we can look at kangaroos. Kangaroos are really good at recording relative humidity and rainfall. And based on the work of Dr. Gavin Perdot, he had looked at auction isotopes from kangaroos that are alive today and found that there are some with low rainfall here in orange, some in high rainfall areas have lower values, and then the ones in medium rainfall have intermediate values. So then we can overlay our values from kangaroo isotopes from Cuddy Springs, and what we find is that it's not going from wet to dry, we're going from sort of a dryish period to drier. So that gives us some characterization that we are indicating, or we are dealing with a period of aridification. We can also actually get at seasonality. So by taking a series of little tiny samples, so these little tiny lines here, these are all individual isotopic samples referred to as serial samples. As that animal is actually growing, its tooth mineralizing, and that enamel is being laid down, it's capturing that signature, the diet signature, the climate signature at that moment in time. And so if we look at a series of these samples over time, we can begin to reconstruct sort of how seasonal that environment was. What was a life of a diprotodont like, for example? What was the climate like that it was living in over the course of a year or two? So what we find at Cuddy Springs is that during the archaeological horizon and the pre-archaeological horizon, throughout time, it was fairly seasonal, and there were no significant changes, but it was a very seasonal environment. Okay, so the other place that isotopes can be really useful is just testing some basic morphological hypotheses. So it was assumed that Macropus was a grazer. Most Macropus taxa today are grazing. That Pertemnodon was a forest browser based on its morphology, and that Stenuris was that some taxa within Stenuris were open country mixed feeders, again based on morphology. But when we look at the isotopes, what we actually find is that while our kangaroo is in fact grazing and our Pertemnodon is in fact browsing, this one that we've presumed to be mixed feeding is actually consuming browse within the densest part of the forest. The other thing that we notice is as you go from the pre-archaeological to the archaeological time period, we see a decline in the amount of C4 resources being consumed by these animals. Okay, so let's look at the whole community. So when we look at the entirety of the community, including things like wombat-sized animals, or wombat-like animals the size of rhinos, like a diprotodont, and a thing called Zygomaturus here. We have different kangaroos, giant wombats, and we look at all of these animals through time, we find that there's sort of a lack of these values here, up here, right? So we see a decline in the amount of C4 resources being consumed through time with aridification. Now this is really interesting because it's not just at Cuddy Springs that we see this. So there was a paper done by Giff Miller and colleagues where they looked at eggshell isotopes and also wombat teeth isotopes and found a similar phenomenon. So prior to 50,000 years, there were lots of periods in which you had more C4 being consumed, whether here or here by wombats or birds. And after the younger periods, you see a lack of the C4 resource being consumed. Now, we don't know if it's that this resource isn't available. or whether these animals are choosing not to eat this resource. But we'll talk about that in just a moment. So what's interesting about this is normally when you have an expansion of sort of C4 resources, things actually do better when there's more C4 resources on the landscape. So for example, in places like Florida, when you go from a glacial period to an interglacial period, you get lots of grasslands, you get lots of happy gomphotheres and mammoths, you get mastodons, you get all sorts of browsers, mixed feeders, and grazers co-occurring together with this expansion of C4 resources. But we aren't seeing this at Cuddy Springs. In fact, at Cuddy Springs, what we think is that these animals aren't actually eating grass. We think that they're actually eating saltbush. So here, if we look at the microwear from modern kangaroos, that are up here in value and that look very, very scratchy compared to some of our mixed feeding kangaroos, our browsing quokka, and our another browsing macropod, the swamp wallaby. And we compare this to all of the Cuddy Springs samples, those in blue and red. They're indicating a much more of a browsing signature, both visually looking at it, but also quantitatively. And so this is interesting. So this means that this resource, this C4 resource that they might be consuming, is probably browse. And so based on some other work that was done in collaboration with Gavin Perdot, and this actually was a fun one that came out of a tea time conversation. So I was at the Australia Museum working on fossils, and he was doing the same, and we were chatting at tea about the fact that Procopton goliath always had a C4 isotopic signal, regardless of what the other kangaroos were doing. And so the other kangaroos were kind of switching back between C3 and C4 in temperate and more arid regions. But Procopton goliath always had these C4 values. And it suggested that they might be eating something like saltbush. Well, saltbush, as the name implies, has lots of salt. And you need water to actually compensate for that high amount of salt consumption. And so when we looked at the microwear, we found, in fact, evidence that Procopton goliath was eating browse, consistent with the saltbush hypothesis. And also when you look at the auction isotopes, they actually have lower auction isotope values than co-occurring kangaroos from the same site. And this also suggests a large amount of water consumption. So putting this all together, this makes Procopton goliath and other saltbush consumers more vulnerable to extinction. Now this can be because they need to come to watering holes and humans may be there to predate on them, or other chompy things like we see here. It could be that these watering holes are fewer in number and also a greater distance apart from one another. And so all of these factors could be contributing. But fundamentally, whether it's humans or whether it's aridification, saltbush consumers are more vulnerable to extinction. So if we put all of this together, some of the things that we can tell from Cuddy Springs is that there was pronounced aridification, that this pronounced period of aridification coincides with a shift in diet away from C4 resources, away from what we think is saltbush, and now you have more animals having to compete for similar resources. Now, while our data do not speak to the role that humans may or may not have played, we cannot rule out climate and subsequent ecological change as a potential cause of some of these extinctions. Now, to say that the megafaunal extinction debate is a heated debate is an understatement, especially in Australia. And this fundamentally comes down to potential different causes. There's human overkill. This can be gradual, or what is referred to as a blitzkrieg. Humans alter the environment, or climate change and other paleoecological factors. Now, it took us a while to publish this paper because of various reasons, as many of us are aware. But within the year that we published it, about a month prior, a paper came out saying humans rather than climate caused the extinctions. Our paper then came out saying, well, we can't say anything about humans, but it looks like climate could cause stress on megafauna. And then about a month later, even maybe a few weeks later, another paper came out saying that there was another site that showed prolonged coexistence of humans and megafauna. So the reason why this is so important is because up to this point, Cuddy Springs was the only mainland site that showed the co-occurrence of humans and megafauna for a prolonged period of time. So this site alone disputed sort of this blitzkrieg idea. And because of that, it was actually came under quite a bit of attack. And so when this other site was discovered, this sort of increased the idea that maybe there was prolonged coexistence of humans and megafauna. And then, within just a few months, another paper came out demonstrating that humans likely came over even earlier. So instead of 45,000 years, maybe 65,000 years. So the reason why I mention all of this is I think it's really important that we don't get polarized into a one-size-fits-all debate when we're coming down to extinction. So humans may have played a role, climate may have played a role, but we need to look at each site individually and also think about synergistic effects that may be happening. So that's why I really like this next figure, which is by Tony Bernowski. It was done in 2004, and it shows that you can have different causal factors in different places. And so much of Europe, the idea is that climate was a major driver, but that humans also played a role. In North America, humans may have been a larger driver, although we're beginning to understand that. And the only place I would actually disagree here with this figure is I would have colored this diprotodont brown at the time for a lack of sufficient data on this. And this is why we're doing a lot of the studies that we're doing, is we're focused on trying to better understand the paleobiology of these animals and potential causal mechanisms as to why they may have gone extinct. So the next animal I want to talk about is this marsupial lion known as Thylacoleo carnifex. I have colleagues who often refer to it as the killer wombat because it is a carnivorous animal that evolved from a group of herbivorous animals that were related to wombats, essentially. But essentially, this was a fierce carnivore. So it has, you can see these two teeth. These are actually two separate teeth. This is not a broken tooth. These two teeth actually make up one sort of shearing facet, so they have these giant sort of scissor-like teeth. They also have a bite force about 80% that of extant lions, despite being about only 30 to 50% of their body mass. And their postcranial morphology is much more sort of bear-like, similar to living bears and things like saber-toothed cats, not cheetah-like. And all of this work was done by Dr. Stephen Rowe, who's now at the University of Newcastle. So based on all of this, we wanted to kind of test some ecological hypotheses and see if we could better understand just the paleobiology and paleoecology of Thylacoleo. And to do this, we looked at specimens from throughout all of Australia, anywhere there were fossils preserved. And we also looked at some other co-occurring carnivorous animals, such as the thylacine, the marsupial wolf, and also the Tasmanian devil. Now again, before we can apply any of these methods, we need to make sure that what we're doing is actually captures dietary information today. So we looked at things like cheetahs, lions, and hyenas, and looked at their microwear. And we know that cheetahs actively avoid bone, and they have values in this range. We know that hyenas actively consume bone, and they have values in this range. And we know that lions are sort of intermediate in between. And so when we looked at Thylacoleo, we find, not surprising, that it's eating everything. It's eating flesh, it's eating bone, it can really do whatever it wants. It's got these massive teeth, it has this high bite force. This all makes a lot of sense. And it's fairly similar to African lions in that regard. Now, what was quite surprising is that Thylacoleo is a specialist of C3 prey. And what I mean by this is it eats things that eat C3 resources. And not just any C3 resources, but C3 resources that represent the sort of the densest C3 resources available. So this illustration paints a really nice picture of what's probably happening. Thylacoleo is hunting ambush style based on its morphology, taking down things like Protemnodon, this forest browser, but it's concentrating its efforts on the animals that are occupying the densest part of that community. Now this is one site, right? So in order to get a better sense of what it's doing from other places, we need to look elsewhere. So we looked at a few different places in New South Wales, the Bingara fauna, and also Wellington Caves, and we see the same phenomenon. We also compare this to other carnivores. What are some carnivorous animals? And so we were looking at the thylacines and the Tasmanian devils, and they're actually eating things from a little bit more open environments. Now we look at this in different regions, and in South Australia, we see the same phenomenon at the Naracoorte Caves. And even in Queensland, where there is an abundance of C4 resources and a large number of prey that are eating these C4 resources, We see an increase in the carbon values that is significant, but still we're in this purple range, right? We are within sort of this C3 vegetation. And so it's likely that Thylacoleo is primarily consuming the densest sort of prey that are consuming vegetation in the densest part of that community. If we look at Thylacoleo from all over, from South Australia, New South Wales, Victoria, Western Australia, Queensland, we find the same phenomenon. So essentially, they are C3 prey specialists. They're eating from forests. So why is this so important? Why is it so important what a marsupial carnivorous mammal that's now extinct did millions of years ago? Well, it provides us with some important cautionary lessons. We think that it was an ambush predator. It could eat flesh, it can eat bone, but it relied on those forests to actually take down prey and hunt within its community. It wasn't a cheetah. It couldn't chase after prey in an open environment. And so our findings suggest that even top predators are really no match for extreme climate change. With aridification, the top predator in Australia went extinct, or top mammalian predator. Okay, so some of the things I've talked about so far are that there is a decline in native saltbush consumers with aridification, this sort of increased competition for similar resources, and also that this marsupial lion can't effectively kill or hunt prey with a decline in forests. So taken together, climate change, including aridification, was a likely driver of large animal extinctions. We can't exclude it. And this includes mammals like diprotodons, giant short-faced kangaroos, and even the marsupial lion. Now, there are other ways in which we can use the fossil record to better understand what's happening today, and there's more practical approaches. So when we build sort of climate models and we're projecting where animals are going to be in the future, we make a fundamental assumption, and that is of niche conservatism. What an animal does today, an animal will do tomorrow. But that's not always true, and in fact, when we try to actually look at these niche models, a lot of this work here looks at the Pleistocene, projects to the present, looks at the present niche, projects back to the Pleistocene based on, say, range maps. And these are only typically effective about 50% of the time, which is, flip of a coin. And this may be because not all animals are abiding by niche conservatism. Some are, so I used to study tapirs, and a tapir is a tapir is a tapir, and they need a forest, and if there's a forest, there's a tapir, and if there's not a forest, there's not a tapir. But there are other species like the spotted hyena, where spotted hyenas were in Ireland. And while we haven't studied those, and I'll say yet, because I would love to, we have studied spotted hyenas from China and compared those to spotted hyenas from Africa. And we actually find that their niche is quite distinct. Those in Africa are consuming lots of bone. Those in China are much more lion-like and not consuming as much bone, and so their niche is actually quite different. So it's important to understand these differences when managing species. Now, another example is that of looking at where these animals were previously. So there's this mountain pygmy possum, very cute, and it is found in Australia's alpine environment. Now, how many of you think of alpine environments when you think of Australia? Okay, there are no hands. And that's because there are very few alpine areas of alpine environments in Australia. Now, what Dr. Michael Archer was able to demonstrate is that at places like Riverslea, there are similar possums that existed in these sort of lowland, wetter environments. And so what he has advocated and worked with conservationists to do is to actually take mountain pygmy possums, rear them at sort of lowland sites, reintroduce them to these wetter sites at lower latitudes, and actually see how they do. And in fact, this could be a really important strategy for thinking more creatively, sorry about that, about species introductions. And in particular, things like reintroducing the Tasmanian devil to the mainland, where it only left the mainland about 3,000 years ago, is another really important strategy. Okay. So another thing that we can do is actually get a better sense of the ecology of animals over deeper time. So this is the quokka, and it is often referred to as the happiest animal on Earth. And it's not necessarily. It has this perpetual smile, which makes it absolutely adorable, but it is highly vulnerable to extinction. In fact, its only distribution is in this region of Australia, including a tiny little island called Bald Island, which is right here, and a tiny little island called Rottnest Island. And actually, about 10 to 12,000 of the quokkas that are still remaining, of about maybe at most 15,000, live on Rottnest Island, this tiny little island. Now, the reason they live on Rottnest Island is because the foxes didn't get there. And so, in fact, they're able to survive in these environments. So we were really curious about their ecology throughout their range on islands, on the mainland, but also in the past. And with the help of Eleanor Schultz, the student I showed on the previous slide, we looked at their dietary reconstructions and see that they're browsing on the mainland, they're browsing on islands, and they browsed in the past. But when we look at the isotopes, we see something very different. The isotopes actually show us that during the Pleistocene, about 30,000 years ago, they occupied more open habitats. So today, when they're found on the mainland, they are in the densest part of those habitats, but in fact, they can and they have survived well in these more mosaic open habitats. Well, this is really important for thinking about conservation. If in these areas on the mainland we can eradicate foxes, we don't necessarily have to reintroduce quokkas to only the densest parts of the forest. They may in fact be able to survive in these more open areas. Now, this has led us to even trying to communicate this message, and so keep your eyes out for a fun children's book that we wrote during COVID that will be out and free online and tells about this story as well. So I'm now going to take you to somewhere completely different. We're going to leave Australia, and we are actually going to go to Southern California to Rancho La Brea, or the La Brea Tar Pits. And this is a phenomenal site, and one of my favorite places to work. I grew up just a few miles away from the site, and the reason it's so phenomenal is because it is a tar or asphaltum seep, and what happens is you have herbivores that essentially get stuck in the muck, and whether it's their distress, their smell, them laying there, they attract lots of carnivores. And so all of these carnivores then accumulate, but they also get stuck. And when they're stuck, we are able to get a high number of fossils from carnivorous animals in addition to herbivores. And this allows us to begin to really look at the effects on herbivores and carnivores, which is so difficult to do in the fossil record, especially when carnivores are often rare. Carnivores are rare on the landscape compared to herbivores, compared to plants, and so much more difficult to study. But at La Brea, we can actually study this in quite a lot of detail. And I've looked at it over the past 50,000 years because we have this chronology. Now, early on, it was thought that based on carbon and nitrogen values, that a lot of these carnivores were competing for similar resources, that the American lion, the dire wolf, the saber-toothed cat, they were all competing for the same prey. So the idea was humans came onto the landscape and are now having to compete with these other carnivores, and they were sort of a tipping point as to why they may have gone extinct. But when we looked at the carbon isotopes, what we actually find is a very different story. We find that the cats, the saber-toothed cat, the puma, the American lion, are all eating things from the forest, which actually makes a lot of sense. Most cats rely on forested prey, with exceptions such as lions and cheetahs, for example. And then the dire wolf is consuming things in more open environments. And then when we mapped on some of the herbivore values, we can see that the bison are right underneath that of the dire wolf, whereas the saber-toothed cats do not overlap at all with the bison occurring from the same aged pits. And the same occurs here. The saber-toothed cats are over here, the bison are over here, and the dire wolves are over here. And so what this suggests is that this idea that saber-toothed cats were just taking down anything in these open ecosystems is not supported. In fact, it led to a new vision of La Brea. And so this was illustration done by Mauricio Antone, and it was based on the data from our paper, suggesting that dire wolves were eating things like bison, saber-toothed cats were eating in the forested environments, things like tapirs, and the coyotes were sort of in between. Now, we spent a lot of time studying the saber-toothed cats and dire wolves, and we continue to study them. And we've tested different hypotheses, whether it was tough times that did them in, what was actually going on. And ultimately, we have been unable to get any answers. All of our hypotheses that we've tested have sort of fallen to the wayside, and we really don't know why these animals went extinct when they did. In fact, they were doing pretty good right up until the end. So what this caused us to do is to actually shift our focus and really focus now on the survivors. Is there something unique about the survivors that enabled them to survive? Are there differences between those that survived versus those that went extinct? And so this has allowed us to sort of begin to focus not on these top predators. So here we've got the giant short-faced bear, the American lion, the American cheetah, the saber-toothed cat, the dire wolf, all of which are illustrated here, and none of these, which are our top predators in North America today, the wolf, the cougar, and the coyote, even make it on this diagram because they are mesopredators or middle-sized predators. So what we find when we compare some of these middle-sized predators to the things that went extinct is actually it was the ability to be highly opportunistic, to engage in things like scavenging that may have given things like cougars the upper hand. So cougars actually eat a variety of different textures. It looks like they are engaged in scavenging in the past, and they do engage in scavenging sometimes today. We also know that cougars, based on modern data, will actually switch their diet based on the presence of other animals. So they'll eat vicuñas and guanacos in South America. They'll switch to eating more armadillos when jaguars are present. They'll eat more deer as opposed to elk when wolves are present in certain regions. And so they can sort of switch and choose based on other predators. And this may allow them to get the upper hand. So secret to surviving extinction, don't be a picky eater. And we thought we'd sort of had solved it. Most of these things that have survived are not particularly picky eaters. So coyotes are also highly opportunistic today. So this sort of makes sense. Well, a little bit, it does, but we're gonna kind of go in a little more depth. So coyotes, interestingly enough, were not opportunistic, were not engaging in scavenging during the Pleistocene. This also makes sense because they had to deal with wolves and they had to deal with dire wolves. Now, the dire wolves were eating whatever they wanted on that landscape, and wolves were kind of being wolves, and coyotes were eating more flesh and maybe smaller animals. And in fact, if we look at wolves for a moment, we can actually test ideas, say, about niche conservatism. A wolf in the Pleistocene, a wolf today, ate similar textured foods, okay? So they were sort of engaged in bone processing and flesh consuming kind of at similar levels as they have in the past. But interestingly enough, if we look at wolves today, we're starting to see a shift away from this. So all of the changes that happened from the Pleistocene to the present, wolves really didn't change their diet, but now they are with modern climate change. So initially, some of our work with Dr. and Bert demonstrated that wolves from Yellowstone actually are eating a higher proportion of, they're more durophagous. They're eating a bit more hard things and more bone processing than wolves from Alaska. Now, the difficult thing here is we had wolves from two different time periods in two different localities. Well, we can't get wolves from the 1960s from Yellowstone because they were no more, and these were the ones we have recently were reintroduced. But we can look at wolves through time in places like Alaska. And what we do in fact see with Ugana Aduba is that you in fact do see a higher amount of bone processing in more recent times. So climate change that's happening now may in fact be altering the diets of these wolves, and more work is needed to follow this up. But the coyotes, we can look at them. They're avoiding bone. They're not particularly opportunistic. We start to see a slight expansion, but really it's in the modern day that they're doing what we see them do today. And in fact, if we looked at things like dire wolves through time compared to the coyotes, it's actually this extinction event that eventually causes the coyotes potentially to sort of shift to their ecological niche today. Now, the other thing that's happening at the same time is the extirpation of wolves. And so we have the extinction first, and then we have the extirpation of wolves once Europeans come in. And essentially, when we look at places in Alaska where you have wolves and coyotes co-occurring, we find that the wolves aren't really the problem. In fact, coyotes will often follow wolf packs around at risk to their own lives, but they do so because they can often feed off of carcasses that they leave behind. And so there is sort of this ability to be a coyote with wolves present, but perhaps not with dire wolves and other predators on the landscape. We can also look at coyotes through time using isotopes, and we find that if we look at these asterisks through time, they're kind of eating prey that are consuming vegetation in sort of moderately sort of open environments, kind of on the fringe of an open and closed environment, but then they really shift significantly in the modern time period. And we think that this is a shift towards the eating of lots of deer. And this could be in part due to the lack of or the decline of apex predators in much of North America, contributing to an abundance of deer carcasses that can be consumed. So all of the work that I've talked about so far is work that we've published fairly recently, but I will put in a little teaser. This doesn't actually include any of the work that we have done on this big project that we're undertaking at the moment, and that's a project called Project Saber. And the downside of La Brea is that these bones are all jumbled together. The upside is we've got tons of them, but again, the downside is they're all jumbled together. So one of the really important things that we need to do is actually radiometrically date every single bone and get as much information from those bones as we possibly can. And so that's exactly what we're doing. We're looking at about 400 different specimens of saber-toothed cats, of dire wolves, of bison, of horses, and coyotes that survive. We're able to get morphological information from the jaws. We're able to get carbon and nitrogen from bone collagen, carbon and oxygen from enamel. We're also able to get microwear, and then we can get a radiocarbon date, and that radiocarbon date can then be matched to a given point in time, which can then be correlated with other proxies of climate. So we can really now start to get a gauge of when the extinction is happening and what's happening with these animals. So this is some exciting work to come. I'm just going to show you one slide of this, which is coyotes showing a dramatic decline in nitrogen after the extinction of the megafauna, or the local extirpation, rather, in Southern California. And this is really interesting. We actually don't think that it's coyote-specific. We don't think it's a trophic shift. We actually think it's a baseline shift in the nitrogen. And in fact, this is supported by amino acid stable isotopes, and also supported by the fact that apex predators today in Southern California have nitrogen values in this range. So essentially what we're seeing is, even though the coyotes survive, they're able to sort of give us some indication of how that ecosystem is changing. And we think that it's changing pretty dramatically after this extinction event in ways in which it's actually depleting the nitrogen from the system, and that's persisting even till this day. Okay. So a few of the things that we can learn from studying places like La Brea are that, highly opportunistic diets might be key to one's success, that coyotes are highly opportunistic today, but they've actually changed their niche and don't exhibit niche conservatism. The thing that they both have in common is that they probably were able to take advantage of smaller prey. So I always like to joke, it'd be neat to see a saber-toothed cat hunt a rabbit, and I don't think they would do so very effectively, but these animals, like cougars and coyotes and wolves, were able to eat smaller prey that did not go extinct. But fundamentally, putting all of this together, what this shows is that apex predators are not immune to extinction. They are some of the most vulnerable if prey decline or their ability to hunt those prey is impacted by things such as aridification. Fossils can also provide us critical insights regarding the ecology of species, and so understanding ways in which their niches have changed over time, places they've lived previously, other opportunities for places that they could live are all critical for management. Now, the last little thing I want to leave you with is that humans are not necessarily as unique as we think we are sometimes. So there was a study that came out and suggested that humans were unique in being sort of this generalist specialist, right? So we're able to sort of be everywhere, makes us a generalist, but we can also be very specialized individually. Now, I agree with this. I agree that humans are one of the ultimate generalist specialists. But interestingly, this got myself and Dr. Richard Hulbert thinking, I think this happens in the fossil record more than we appreciate. And in fact, when we looked at this one horse that we had already been studying, we saw that it had a very wide breadth of isotopic values. Some individuals were browsing, some individuals were grazing, some individuals were mixed feeding. But when you zoomed into any one individual and you looked at their serial samples, what we actually found is that they were individually specializing. They weren't eating grass at one interval and browse at another. They were eating just grass, or they were eating primarily browse. And this got us thinking, are generalists composed of individual generalists, or are they actually composed of individual specialists? And with this team of folks, we set out to sort of answer this. And why I think this is so important is it's so critical to how we actually manage populations. So when we looked at another site, we noticed that this camel, even though it switches from sort of being more of a browser during a glacial period to more of a mixed feeder during the interglacial period, at any one interval, or if you zoom in on any particular individual, they show very little variation. And so again, it's a series of individual specialists. Well, then we broadened this out even more. We looked at numerous individuals across the globe. In fact, we looked at all mammals, all herbivorous mammals that we could look at that were published. We sampled some as well across the globe over the past 7 million years and categorized them into different dietary categories, and essentially find that individuals, whether you're a generalist or you're a grazer or a browser, more of a specialist, everything is individually specialized. And again, if we sum this up in sort of this sort of a plot which looks at the total amount of isotopic variability, we see that so our mixed feeders, which is very surprising, have low variability, but everything does. Now, why is this so important, and why did I choose to share this as part of this presentation? I think it's something that we often don't think about. We think of species, sort of individuals, occupying the same exact niche of the species. But in fact, in the case with generalists, in the case of many species, you have them made up of individual specialists. And so this is really important for thinking about management and thinking about making environments that are diverse, available to different mammals on the landscape, to make sure that they are able to become and be the jack of all trades and masters of all that they are. It also provides us with an interesting understanding of evolution and how generalists may have survived in many scenarios. Okay, so I've talked quite a bit about climate change, including aridification, some of the drivers of potential large animal extinctions. We've talked also about the importance of apex predators and not assuming that they will fare well. In fact, they are so key to keeping a healthy ecosystem. In fact, they are responsible for making sure that the prey are not overly abundant and then don't wreak havoc on the ecosystem. But yet they are not immune to extinction. Additionally, the fossil record can provide us with some critical insights regarding the ecology of species before European arrival. It can also clarify ecological niches prior to the Anthropocene. But the point I want to end on is this. Our past provides us with critical data, and as many paleontologists are in the room, I know we are all committed to understanding the past. But our past provides us with this critical data that we can use to make informed decisions. However, we can only improve our future if we act on this knowledge. And so with that, I just want to thank all of the following individuals. I couldn't possibly list all of the people who have contributed to all of this work, so I chose to instead post students, past and present, up here on this slide. I wanted to thank members of our dream teams, numerous museums. The work featured here is mainly from the Australian Museum, the Florida Museum of Natural History, the Brea Tar Pits, Queensland Museum, South Australian Museum, Santa Barbara Museum of Natural History, and the Western Australian Museum. I also want to give great thanks to the Royal Irish Academy for having me here today, the Paleontological Association as well, and also Dr. Maria McNamara for all of her organizing and planning of this event and this conference. I also want to thank my family and friends and colleagues in a variety of ways. So thank you so much. And very lastly, I need to thank the important funding sources that made all of this work possible, including Vanderbilt University and the National Science Foundation.
Well, how do you follow that? You don't put up lots of slides. No, at its simplest, searching for and studying fossils tells us something about which animals and plants were alive at various times in the past, where they lived, how the present-day organisms got where they are, and where they've evolved from. And then depending on the preservation quality of the fossils and the numbers in an area, we can get some idea of the species richness in that area and of the nature of the species composition and the community structure in the past within that area. And then, as you've seen, based on comparisons with present-day relationships between similar kinds of communities and their environment, paleoecologists can hypothesize what the climate was like in the area at the time the fossils were deposited. But ecology is more than just numbers and types of species. It involves interactions between organisms and with their environment, both the abiotic physical and chemical environment and the biotic environment, the other species, the competitors, the predators, the parasites. The fossil anatomy, or even information about the colour of fossil animals, allows paleontologists to draw some inferences on animal behaviour and speculate on these interactions. But as Professor DeSantis has pointed out, studying the diet of animals is important and much more direct. Identifying animal diets tells you about the species interactions, such as competition and predation. It can inform you about population biology. It's essential to inform the construction of food webs, identifying all the feeding linkages within the community of the habitat. And it can also illuminate very important ecosystem processes like the flow of energy through the community and nutrient cycling within the ecosystem. An animal diet, as we've seen, is susceptible to changes in the environment. And changes in the presence or diet of key species in the community can cause a cascade through the food web, which can in turn influence the whole ecosystem. So how do we investigate the ecology of the past, the interactions between species that occurred long ago? How did these ancient organisms interact with their environment and adapt and evolve as the climate changed? And if we can understand what happened in the past, how can we use this information to try to predict what might happen in the future in our changing world? And paleoecologists and paleontologists can look back thousands and thousands of years to piece together what the ecosystems of the past look like. For example, you can go to a lake and you can take a sediment core from the bottom of the lake. And in that sediment core, you can find pollen that blew into the lake from the surrounding terrestrial vegetation. And what's good is that sediment core is like a clock. The deeper you go into the sediment, the further back into time you go to when the sediments were deposited. So looking at the pollen, you can piece together the changing vegetation in the landscape surrounding the lake. And that in turn provides information on regional climate at different times in the past, as well as evidence of human activities in the area around the lake, like deforestation, for example. And looking at microfossils, small crustaceans, small diatoms, small plants found in the sediments can also inform us of the lake ecology and the past climate and stresses over time. Another interesting example of what ancient ecological artifacts can tell us about the past is from fossilized piles of feces, or middens as they are called, of the rock hyrax, which is a very interesting rabbit-sized herbivore which is common throughout southern Africa. And they have a habit of defecating in the same place over many generations, like a communal latrine. And you can excavate into these middens, and you can analyze them for environmental proxies like fossil pollen, like stable isotopes, and biomarkers like environmental DNA. And when it's linked with high-precision chronologies, these proxies can provide a record spanning upward of 50,000 years of the past ecosystem and of climate change. But how do you identify other species interactions in the past? And especially what different kinds of species eat and what eats them? Now, if you're a present-day ecologist, the best way to identify diets is to observe the animals in the lab or in the wild, or examine their gut contents, or examine their feces for remnants of prey. Now, where that's not possible, ecologists will use various biochemical and anatomical proxies to provide generalized information on the diet. And we use tools like the ones you've seen, stable isotope, carbon isotopes, nitrogen isotopes, to identify the diets of the organism in the present. You can tell what trophic level the species is feeding at. Is it herbivore? Is it a primary predator? Is it the top predator? You can tell what the main carbon source was. Is it from plants? Is it from dead organic matter? Is it from other animals? And what Professor DeSantis has shown in this wonderful talk is how this approach can be extended deep into the past. And using the powerful analytical methods that she's given us a glimpse of, not just the diets and the ecology of species in the past, but also what this tells us about the environmental conditions then and how individual species responded to them. Now, historians very often advise us to learn from the past, and we can apply the same tenet to ecology and to climate change. Current climate change is like an ongoing but uncontrolled experiment. And whilst we've got a good idea of what changes might happen in the future from modelling and the like, we don't know for certain what the effect will be on our current ecosystems. But using the techniques Professor DeSantis has described opens the window to the past in such a way that we can begin to see the impacts of change over time, like an experiment in the past. And we can use this to make more informed predictions about what the current effects of climate change might be. And it's not just related to the dramatic possible species extinctions, but also, as we've seen, to more subtle changes in species interactions, and even finer adaptive changes to the ecology of individual species. It's truly fascinating science. And these explorations into the past can help us assess how adaptable individual species might be to environmental challenges like drought, how resistant ecosystems and their processes might be to climate change, how quickly these ecosystems might recover if the changes are reversed or stop, and whether there's a tipping point beyond which recovery to the previous configurations of species is not possible. They also, as Professor DeSantis has shown, give us pointers that can help in species conservation going forward. So, thank you, Professor DeSantis, for such an excellent discourse, opening our eyes to the past, showing us how this can potentially help conservation to the future. There's also a very strong message that's worth repeating, especially given what we're seeing today in relation to the effects of climate change. Knowledge of the past can only improve our future if we act on it. Thank you.
You can all hear me? Right. We're going to take some questions from the floor to Professor DeSantis. Anybody who would like to, we've got a roving mic here, and perhaps we could see some hands. Yes, the lady in the green jersey down at the back. It's Maria. I should be wearing my glasses.
Can you hear me?
Yeah.
So, Larissa, I was thinking about your camels and the fact that they're not real generalists, but they're a load of picky eaters. And so I guess, the key message here in terms of conservation is that we need environments that have diverse food plants and diverse subhabitats to allow mammals to have this sort of individual specialization. Is that the kind of key conservation message?
Yes, absolutely. So that's one of the primary messages, is that we really need to, diverse ecosystems are beneficial. We also can't necessarily generalize what a species does to each individual. And we see such different levels of niche partitioning, whether it's at different sites or localities or places. And then even within a population that's highly generalized, even those individuals are doing things differently. We're very curious as to why this is, and it may be a way of sort of reducing competition with each other and also amongst other species. And it may also have to deal with certain constraints like gut microbes and things that may play a role. But ultimately what it means is we need to really think a little bit broader. If something is sort of a mixed feeder or something is eating a variety of different resources, we need to allow it opportunities to be able to just graze or just browse or mixed feed in these different habitats. So mosaic habitats may become really critical for a lot of these species. Yeah, absolutely. Thank you.
Very much. Yes, gentleman down there. Yeah.
That was great, Larissa. Thanks for the talk. I just wanted to ask if you could expand a little bit on what you mentioned about the synergistic effects between climates, human interactions. For example, is there any evidence that sort of pre-industrial human deforestation may cause local climate changes? It may cause aridification just because of human interaction with the vegetation rather than direct hunting impacts.
Yeah, yes, absolutely. So, often my students will actually ask a very similar question when I talk about megafaunal extinctions. They basically say, why does it matter, right? Why does it matter if it was climate change or whether it's humans or human hunting or habitat fragmentation? And what I typically answer is this, for the long time, especially in places like Australia, we focused in so much on human killing of megafauna. And it's been through a particular lens, a particular viewpoint that people have focused on this hypothesis. But really, what we need to do is understand, maybe humans played a role, but did climate play a role as well? Because now we're living in a world in which we can't disentangle the effects of humans, habitat fragmentation, climate change. They're all happening. So if it wasn't just human overkill, which it doesn't look like it was, then we really need to think more broadly about the world in which we're living and the impacts we're having, and that presumably in a world in which human overkill killed the megafauna, And that's the only reason. If we were to stop direct hunting animals today, then everything would be hunky-dory. But I don't think that's the case. And in fact, places like Australia, I think climate and aridification really did wreak a lot of havoc on these animals. In fact, a lot of the browsers just disappear. And so where are they, right? And they're not there. And we have someone in the audience who's talked quite a bit about browsers disappearing, but that can have quite an impact on these ecosystems. Additionally, in places like North America, humans may have had a larger role than climate or a synergistic role, and we really need to evaluate all of those hypotheses. And again, today, it's all of the above. And if all of these impacts are having an effect, let's figure out the best way to actually manage these populations moving forward.
Thank you. Karen, we have a gentleman here.
Thank you. Just going on from what you just said there, in Ireland, a small island, we can actually see, I'm 69, and I can see as going to national school, it was the countryside, even the agricultural part of it was like a zoo. It was incredible. And I miss that all the time. And it's getting worse because it is human effect. It is like agriculture is 80% of the country really, and we'll say forestry is 10%. The forestry is by and large monoculture, and when you plant the conifers, you're knocking out the ground underneath. Nothing lives there for 35, 40 years. So like we know the issues here, and we know what's to be done, but like it's going to be a huge challenge.
Thank you. And Karen, I think there's just a gentleman behind you there, and then you might take those two together.
Perfect.
Larisa. Yeah.
What was the cause of the baseline shift in nitrogen availability in California that you mentioned?
So that's an excellent question, and that's actually something we're working on at the moment, and we have some hypotheses. And I'll be excited to share it, hopefully in a few more months when it's ready to be published, and I can talk with you more individually as well. But we think that the ecosystem is changing in a pretty dramatic way. Again, some possibilities include decline of megafauna, changes in nutrient cycling. Others can include, human involvement, changes in fire regimes, for example, and that some of these different effects may then be causing a change in nitrogen. So it's hard to know the exact cause, but we can try to eliminate different hypotheses, and that's what we're sort of in the middle of trying to do. But ultimately, what it shows, and what I think is really compelling, is that you have this extinction that happened, over 10,000 years ago in North America, that's still having an impact today in this region. And it's also sort of a cautionary tale of what we're seeing happening in the region right now. And absolutely, I just wanted to also agree with, islands are much more vulnerable. They tend to have lower species richness. Invasive species that come over can also wreak havoc, and habitat and land conversion is a major can have major impacts. In fact, one of the things that we're currently studying as well are thylacines on the island of Tasmania. And what we actually find is thylacines, they were doing pretty well in what they should be doing morphologically, eating lots of flesh and things. But once Europeans come over to Tasmania, and we think not only are directly hunting them, which we know was happening, but also converting the land, that they're also having an indirect impact, and they're causing a lot of the thylacines to actually have to scavenge. So we're starting to be able to pinpoint consequences of our actions on a finer scale, but absolutely, whatever we can do to sort of maintain and retain biodiversity that exists today is absolutely critical.
Anybody else like to ask a question? Yes, right behind me here.
And we see it, incidentally, in Australia, where there is introduction of rabbits and cane toads. These wouldn't have come along with the initial Europeans, as it were, but at a much later date.
Right.
Is there any incidental variation in new species during the periods that you looked at that might have actually resulted in the changes?
So maybe you can clarify just a little bit. I don't think I'm fully understanding the question.
Was there any new invasive species at the pivotal points? So let's say, for example, like we have mice at the moment in Australia.
Right.
Yeah.
Yeah, so I mean, I think foxes being brought over was a major one. So there's a few different phases of invasive species that, invasive species can be a natural phenomenon, and we look at, for example, North and South America. South America was isolated for many millions of years, and then the Panama Canal connected the two continents, and you had this sort of mixture of different species, which had profound consequences on the flora and fauna in those regions. In Australia, you have a variety of things happen. You have dingoes come over a bit earlier, right? And then you have eventually when Europeans get there, things like foxes are brought over. These have really wreak havoc on the ecosystem because they can predate on a lot of these native fauna that haven't had to deal with foxes in their ecosystem. And so we can start to see some downstream consequences of that, and then later things like rabbits and cane toads. And so, it's not a wonder that, island nations are very careful with quarantine and trying to make sure that foreign pests and things don't get onto their land, because they can cause really profound impacts on the biodiversity. And so it was interesting to sort of see how things actually played out even with COVID, because coming from working in Australia and seeing how seriously they take quarantine, we could then see how seriously they took quarantine sort of under the pandemic situation as well. And when you're an island nation, your biodiversity is much more vulnerable to extinction because of that isolation for so many millions of years.
Paul, I think you had some follow-up.
Thanks, Mary. It was interesting to see the extinction of the top predators. In the fossil record, do you see any evidence of this cascade that can happen in the community when you take out a top predator, like you can see in lake fauna, for example, in the present day?
Yeah, absolutely. So we largely think that, the coyote and wolves have sort of, and also cougars, have become these apex predators in North American communities when these predators go extinct. And so you basically you can see changes in body size, potentially. You can see changes in their diet in some cases. And even, so I live in Tennessee. We don't have wolves anymore, but we have coyotes. And our coyotes have sort of become these sort of apex predators in the ecosystem. They're actually eating quite a bit of deer and deer carcasses from automobile accidents, and some that they may be naturally taking down as well. But we're seeing a real shift. And so I think that's really important point to make, and the point also made by the other gentleman about baselines and watching ecosystems change, is that we see these megafaunal extinctions, we see shifts in some of these communities, but what we're seeing today is a depauperate fauna compared to what it has been in the past. And what each of us see in our lifetime is going to be an even more depauperate fauna than our ancestors would have seen. And so I think this is really important to consider moving forward.
Thank you. We've got one last question down here. Oh, sorry. There's obviously two more. I'll allow this one and one more, and then I think it's probably time for us to wrap up. So go ahead.
So we've seen a lot of these apex fauna in Australia and North America, but not so much in Africa. So what's going on in Africa?
Yeah, that's a fantastic question. There have been some megafaunal extinctions happening there. One idea that has been proposed is that because humans largely became that humans were not novel predators in Africa like they might have been in other parts of the world, and essentially that some of the predators were able to, predators and prey and ecosystems were able to function. That being said, this is still being tested. There is some examples of some megafaunal extinctions, but it is far less on Africa than it is in the rest of the world. And we actually see these extinctions happening at different time intervals. They don't all happen at the same time, and they largely do happen shortly after human arrival. Now, what that period of time is varies. So it may in fact be, in many of the cases, that the climate is changing and you have now humans that are efficient predators, and they may be different levels of efficiency on different continents at different times by the time they get there, different technology. But essentially, you have these efficient predators that are perturbing the system that is now being affected in some cases by climate change, in some cases it may not. But these two things together are synergistically providing sort of a tipping point that's causing the decline of a lot of these animals. And so that tends to be what at least I'm seeing when we're looking at places like North America. In Australia, I haven't seen any clear evidence of human contributions yet. Doesn't mean that's not the case. We see more evidence that climate was a phenomenon, but we do know that humans have wreaked havoc on many ecosystems across the globe. So I think it's really important to, when looking at sort of these extinction debates, is to sort of keep it local, keep it regional, and really better understand what's happening in that system, because it's not a one-size-fits-all, and in fact, it's fairly complex and not as simple as we've been sort of breaking it out into to be.
Okay, and one last question, Karen.
Hi, thank you, Larisa, for this wonderful talk. So can I ask a question about the generalists being individual specialists? So I'm just wondering, how common do you think this strategy is in the animal kingdom? Because like niche petitioning among different age groups is pretty common in the animal kingdom. So we see them in fish, in reptiles, mammals. But what about these individual specialists? How common do you think they are? Thank you.
Yeah, that's a fantastic question. I don't think we know the answer, but I think it is fairly common. I mean, we looked at all herbivorous mammals that we could look at, where we had teeth available to test this. People have studied this in different ways, looking at carnivores. So a lot of the work that has had been done, folks like Dr. Hannah Vanderzayden had looked at different sea turtles and had seen that, even within a sea turtle community, a modern community, they can be individually specialized within that population. And this makes a lot of sense in the case of carnivores, right? Because your search picture for taking down prey might be very different. So if you're trying to hunt a rabbit versus hunt a bird, how you hunt those animals is completely different. And so you may then specialize on birds or lizards or rabbits more than, say, another individual does. But in herbivores, we really didn't know or think this was happening. The reason we were able to figure it out is because we use these serial samples in the fossil record to get at sort of changes in environment, or how have horses changed their diet, or how has the climate varied throughout a period of a year. So we've used it for other purposes, but then when we pulled all of this data together, which is thousands of specimens from all across the globe, everywhere from I could name off, I don't know where to start. There's specimens from Ireland and from China and from North America and from Brazil and everywhere. We see the same pattern. Now, there are a few individuals that do show higher variation, and those tend to be grazers. We've seen it in some things like mammoths. We've seen it in wombats. Seen it in bison. And so what this shows is that it is possible for some individuals to sort of vary their diet more. The method is likely picking up on this. It's just very rare. And I do think that it's really important that we go back to studying a lot of modern animals today to better understand this phenomenon, and in fact see how common it is amongst other groups of organisms, not just mammals, but other things, because it could really change the way in which we think about managing species, and it definitely changes the way in which we think about sort of evolution and also scaling up. An individual doesn't necessarily have all of the aspects of its species. It can be specialized, and sort of better understanding this at an individual and population level. So yeah, I think there's a lot more work that can be done in this area, and I think it's really interesting that this is a phenomenon. But I will say it makes a lot of sense, right? So, the fact that the trade-off of being, if you're a generalist, you're a master of all, I mean, you're a jack of all trades and a master of none, right? So you can do a variety of things, but none of them particularly well. And this actually gets away from that constraint because individuals can be individually specialized. They can be masters of all. So if the climate or environmental perturbations happen or competition or whatnot, it kind of buffers individuals from extinction in some cases. So it makes a lot of sense, but I think we need to do more work on it, absolutely.
So it just remains for me to thank you, Professor DeSantis, Professor Giller, for a wonderful evening. A couple of points. So what we've learned tonight is that looking back into the deep past through bones and animals, we can learn a great deal about ourselves now. And in a week that the Webb telescope taught us as well that we could look back and back into the deep space and not understand our own existence, this is a time of great challenge. And I think some of I think that the value of science tonight is really coming to the fore. So as a humanities person, I have to say I'm humbled by this, and I want to thank you all. I want to thank you for the wonderful questions. I want to thank the Secretary of the Academy, Mary O'Dowd, for organising the Academy's discourses, and I invite you to join us online for our discourses. The next one will be on the 8th of September, and it's more history, but it's a different kind of history. It's a panel discussion entitled 100 Years on Ireland, Finland, New Zealand and Poland, and it's a comparative history. The details will be on the Academy website.